give you a bit of background on the project, this project is in collaboration with a couple of guys that are looking at tuberculosis granulomas. So they have treated and untreated subjects where you can see the tuberculosis granuloma severity getting worse. So you have an inflammatory type of granuloma versus a more senescent fibrotic type of granuloma. And they're going to take the granuloma borders and try to see what kind of immune cells localize in the inflammatory versus the fibrotic components.
So this will be a lung tissue most likely, right?
Yeah.
Okay. So let me pull up my presentation. Okay, can you see my screen?
Yep.
Okay. So the instrument that you will be using is Lionheart. We also have one more instrument, but that's not really set up right now. It's going to be easier to use that in future versus Lionheart. So just kind of letting you know about that. So Lionheart is going to be really good for color imaging of H&E samples. It can also do epifluorescence imaging for immunostain samples. We will also be doing part of epifluorescence on your slides, and I'll show you exactly at what stage that comes into picture. And Lionheart has the ability to talent stitch the tissue or even large field of views, because if you have a large field of view, you really cannot look at the entire area, and that's where talent stitching function of Lionheart comes into picture. So talent stitch, what I mean is you acquire individual images and then you stitch the entire area. And one of the advantage of that is you can zoom into a particular cell and it still looks proper cell versus bunch of pixels. If you were to image something at using, let's say, a 4x magnification and you keep on zooming in. So steps during Visium pipeline where we really contribute to your project. So the first step is to determine the optimal time for tissue permeabilization. So Visium is going to give you 2 slides. One slide on which you have this 8 different squares on which you will put your tissue. One of the square is going to be a positive control. The second square is your negative control. And for the rest of the other squares, you will end up looking at, you will end up putting your tissue and then look for the permeabilization time based on the binding ability of the probe. And the second step is actually where you put the 4 tissues, as you mentioned, and we will do H&E imaging before the tissue goes into library prep. Okay. So the reason for doing H&E over here at step 2 is to make sure that the tissue integrity is okay and you have a proper tissue onto your slide, as well as overlay the final readout of your mRNA copies onto a particular cell per se. I mean, I wouldn't say a particular cell because the region that you are spatially looking at is about 50 microns in size. So you don't get a cell, but kind of a 50 micron region per se. Okay. So in the first,
the tissue optimization slide you had in the, there will be no imaging, well, there will be some imaging done on that as well, right?
Yeah, so that's what I'm going to. On that slide, we'll be doing epifluorescence imaging at that, let's say, 568 nanometer wavelength.
Okay.
So pretty much like a Texas red or TRITSI kind of imaging over there. So for the tissue optimization slide, this is how the slide looks like on the left. These are the capture areas that you have. One of the capture area is your negative control. The second capture area is your positive control. And you have this other 6 areas, capture areas on which you will also have your tissue. We will do an H&E stain first to make sure, an H&E stain and then image also, to make sure that the tissue integrity is perfectly fine. And once that is done, you will be doing the second step, which will be visualized using the epifluorescence imaging, typically using a Texas red filter set, in which the mRNA that you have will be fluorescently labeled. Nucleotide will be binding to the mRNA, pardon me, the fluorescently labeled nucleotides will bind to the reverse transcribed mRNA, essentially to know whether or not your tissue is good enough. Okay. So let's say, for example, when you're looking at step 1A, which is the H&E staining for tissue integrity, we are looking at this step right now, 1A. This clearly shows that your tissue is perfectly fine. And then when we do the permeabilization step and we image that, so the negative control should look completely blank. The positive control will be super saturated because there is a ton of RNA present over there. And this is the critical part. Probably if you had talked to Fred or somebody, they would have told you that you need to determine the tissue permeabilization time, and the tissue permeabilization time varies between the tissue. Lungs will be different than liver, brain, heart, and so on. So every tissue will have a different permeabilization time. And for you, it's going to matter a lot since things are getting shipped from Texas. So that is another variability that you need to consider. So let's say, for example, in this situation, the optimal tissue permeabilization time is either 18 or 24 minutes, because when we go through here, 3, 6, 12, we don't see much of the nucleotide signal, versus when we go from 18 to 24, there is this decent signal that we are looking at. So you would choose either 18 or 24 minutes in this example. Okay. So once you have selected that, we can also kind of pinpoint which areas have higher mRNA signal per se. So we can overlay your H&E stain with the nucleotide stain. This is in case if that is of your interest. And second step that we are looking at, this is exactly what you end up getting, right? So we will image each of the 4 capture areas. These are no longer capture areas, but individual regions where you have the tissues. And once we zoom into one of the, yeah, go ahead.
I had a question about step 1. The positive control, what tissue is that? Is that something that I need to get a hold of myself, and
no, you
something
don't need
with a
to,
lot of RNA?
Yeah, you don't need to get a hold on of anything by yourself. The slide itself, so over here, the slide itself will have this region with a ton of mRNA.
Okay, but the bottom 6 squares, there's nothing. The tissue will have to be loaded, right?
That's correct. So bottom 6 square as well as this, the tissue will have to be loaded.
Okay. So the top right is, and I thought that was the negative control. No.
So the top, exactly, the top right is the negative control. That's what you are looking over here, right? So top right, if you flip it, I haven't flipped the image, but if you rotate this 90 degrees towards right, this becomes your negative control. This becomes your positive.
But when you say negative control, it's just the permeabilization is 0 minutes. Is that how you define it?
So you don't, the way you define it is you don't add any of the fluorescently labeled nucleotides that bind, right? So even if, that is just to know the background of the sample.
Okay.
Okay.
So, but one more question is, so if I were to get the samples from Texas, I would ask the collaborator there to load everything except the positive control and put it on dry ice at the same time, right?
That's correct. So if you get hold of this slide when you buy the kit, uh, things will be clearly marked. I cannot see things properly, but I'm pretty sure that things are clearly marked as to what capture area is a positive control area, which one is negative control area. So it will be clearly marked.
Okay, thank you.
Okay, yeah. And the other thing I would advise pretty strongly is before the folks end up, let's say, putting any of the tissues over here, I would recommend you guys to create this 8 by 8 kind of a grid of the similar size on a regular slide, and try to put the tissues the same way, because um, it's okay to put one of the cryosection slice onto a slide, but it's a different ball game when you have to put all of them in a particular area. So that's a bit of challenge. So I would definitely advise people at Texas to practice this on a simple slide before spending their energy on an actual Visium slide, because once you make a mistake and you go and ask the Visium folks, they will be like, oh, you know, you have to pay for it. So just trying to save you some money over here.
Okay.
Okay. All right. So again, kind of rehashing out step 1A, just to make sure that your tissue is perfectly fine. Step 1B, to figure out what is the permeabilization time. And this is the permeabilization time that you will end up using when you do step 2 for the library prep, right? So then we will take the images. I typically use a 20x objective and acquire 2 of these areas at the same time. And it's important for us to capture an area which is outside of this circular dots a little bit. And though it looks like kind of small to you, but when we do tiling and stitching, we can in fact zoom in onto a particular cell, right? So we get good enough resolution. And with the limitation of Visium, you are essentially looking at a 50 micron area when you look at the nucleotide signal at the end. Okay, so in terms of my personal experience, it takes about 50 minutes to acquire a 20 by 20 tile image, which is what you have over there. The data set that gets generated post stitching is big. It's roughly about 1 gigabyte in size for each of the image over here that you are looking at. In spite of this long times, when I helped Vasanta, there was no effect on the library preparation or the quality of the RNA. That was also perfectly fine. And this is something which is different. Now, Visium also has immunofluorescence-based validation, so you can ignore this part, okay? So it now really depends because this is, there is a collaboration between Texas and Emory. It's really important to maintain the viability of the tissue and make sure that whatever time that you get in terms of permeabilization, nothing gets skewed between this step and this actual step. Okay, so if there is any change in the handling process, the permeabilization time will differ when you are at step 1B versus when you're at step 2. So that is something which you will have to take care of.
So if I were to, should I do the optimization staining first and run it by you, and once that is ready, then thaw this second slide and go through everything? Or do you prefer that we do both slides at the same time?
No, definitely. You would do this in 2 different successions. So you would do the first step, which is, and if you look at the protocol, you will kind of get an idea where in the first step, you will simply do H&E staining, and then post H&E staining, you will do permeabilization. We will image these slides for the optimal signal. And not only you will run this through me, but you will also run this through 10x people. they are always very happy to give you feedback of what they think about your tissue and about the permeabilization time. So definitely run it through Fred and Egon. Egon, more than Fred, Egon is involved heavily when it comes to giving you feedback of what looks right, what looks wrong.
But how long do you think it typically takes then to go through the optimization slide and then move on to the next one? It's a day-long thing.
So this is so step 1 is a day-long thing, and once we know the permeabilization time, then you can start your step 2.
Okay, so if so, I'll have to book time with you guys 2 days in a row, right?
Exactly. And Laura will take care of that. So if you let us know what days you want to perform these experiments, Laura can block those times on Lionheart and help you out.
Okay, sounds good.
Okay. So let me just stop sharing so that I can see you. Okay.
Okay, and so the next question was about the cost, because we want to put this is just a pilot run. We want to do that. We want to put this into a grant and then propose a budget in the grant for a much larger number of samples. So how much do you?
Yeah, so in terms of cost, there is subsidy. I don't know if you're part of pediatrics or School of Medicine or what, but depending on your affiliation, there is subsidy. And we will charge you, and I can send you the link to the page where we have our costs, and you can figure out how much you'll be paying at the end. And because Laura will be doing this for you, it's going to be a facility scientist project in a way. In terms of total number of hours, it's going to cost you, let's say, about roughly around 5 to 6 hours of total time. Yeah.
Okay, sounds good. I'm with pathology, so just to let you know, I started as an instructor in the pathology department at the end of October. So
okay, wonderful. I'll send you the link of the costs, and also you can figure out exactly where you fit in that link. Okay.
Okay. And the last thing is, can you also send me this recording or the presentation, either way?
Absolutely, I'll send that to you. No worries.
Okay, thanks a lot.
Yeah, you're welcome. Anything else, Ashish?
No. So what I'll do on my side is I'll let you know when the tissue comes in and I've validated the RNA integrity of the bulk tissue. And when I let my collaborator know, I'll send in both the optimization slide and the other slide. And whenever he's ready to ship, I'll contact you and you can put me through to Laura.
Perfect. And even before all of this, please ask your collaborator to practice putting the cryopreserved tissues on a simple slide by drawing like 8x8 grid or essentially 8 squares on the slide. Because from my experience with Vasanta, that was challenging. It's not a trivial thing to do. So I don't want your Visium slides to get wasted in no way.
Okay. And in general, if you have done the optimization step for a specific type of tissue once, you ran another experiment, 2 weeks from now and then another 2 weeks, do you do the optimization step every time? Or
so from what I have learned from Vasanta, here's the deal. If you are working with human tissue, there is a lot of variability in the permeabilization time, and that variability really comes from the inherent process of isolating the human tissue. So which is the process that you use to get the tissue out of humans. Let's say you do endoscopy, right? And you take the tissue out. And that process itself can damage the tissue to an extent. So the permeabilization times were different for her, let's say, liver tissue that she took out. But that's part and parcel of the process. But if you're working with animal per se, let's say mouse, then it's not a big deal.
Okay, sounds good. Thank you.
Okay. Yeah, you're welcome. So I'll send you the recording.
Okay, I'll talk to you later.
Take it easy, Ashish. Bye.
Bye.